My name is Ocheltree. I'm an assistant professor in the Forest and Rangeland Stewardship Department. My junior year as an undergraduate, I had a plant biology course from a professor who was a very engaging kind of professor and really asked lots of kind of hard problem-solving type questions, but they were all about plants. And so I think partly because of his teaching style, but also like the subject matter kind of came together, and I really enjoyed the way of plant biologists thought and the topic, and so I decided at that point that I wanted to study kind of plant biology, plant physiology. The biggest problem that we're trying to solve right now with our work is figuring out how many individuals in a plant community might die during a drought, so that once that drought is alleviated, we can predict like how much grass is going to be there, how many plants are going to be there afterwards that might provide forage or habitat for wildlife. The most rewarding part of my career thus far is just really all of the interactions that I've had with various students. The questions that they ask, their curiosity, just those types of interactions and helping them discover those things that they're interested in and they're passionate about is just really exciting, and I always come away like feeling really like great after those kind of experiences. So I'd have to say those are the highlights.
Hello everyone. I think this is our last presentation of this whole semester. So welcome to you all. So my name is Khun Thapamagar, and I'm a PhD student in Rangeland Ecosystem Science under the supervision of Seth Davis. Today we have presenter Dr. Troy Ocheltree in our seminar. Let's talk a little bit about his biography. He did his PhD from Kansas State University in agronomy, and he did his MS from University of Idaho and BS from University of Minnesota. So in research, he is broadly interested to understand a plant response to the environment, and his lab specifically trying to identify the mechanism underlying drought tolerance strategy in plants from cell to the rangeland and also like continental scale. So with that, let's join to me to welcome our speaker, Dr. Troy Ocheltree.
Thank you, Khun. All right, who came up with an answer that they want to share with the class? I know, this is a horrible place to like talk and share.
Is your lab allowed to respond?
No. Miranda.
I'll answer. So drought tolerance we discussed is being able to tolerate really low water levels.
Okay.
Like handle low drought conditions and still survive. Drought avoidance would be being located in areas that don't experience that high water stress levels.
Okay, so you're avoiding those conditions altogether. Perfect. I'll come back to this a little bit later in my lecture, so that was the thing I thought we could all talk about and have an idea about, and it will tie in a little bit later in my presentation. So I'll come back to these different ways of handling drought. But I think of both of these as being ways to resist drought or to grow in a place where we have drought conditions. And they're mechanisms that we've kind of known about for a really long time. And I think we're really just starting to incorporate them a little more in our understanding about ecosystem processes. And so I'm going to show some data today that hopefully kind of convinces you of that. So this is my title. I'm horrible at titles, even when I know exactly what I'm going to talk about. And when I gave the title, I didn't know what I was going to talk about. So you might have read this and said, like, what the heck is he going to talk about? Like, from cell to continental. That was just my way of encapsulating anything that I wanted to say today. So, but I'm really going to talk about how plants, what are the mechanisms that plants utilize to survive and grow during drought, and what does that mean for the productivity of ecosystems and us thinking about managing them and utilizing those natural resources for their valuable ecosystem services. So before I get into it, I just wanted to acknowledge most of the people, hopefully, that have had some impact on either the ideas I'm presenting or some directly involved in the data collection. First, lots of graduate students. These are ones specifically from my lab. Oop, forgot to finish Crystal's name. But these are lab students that either have worked or are working in my lab now. I've been lucky enough to interact with students from other labs on campus. They've done work in my lab, or we've collaborated on something. And I've had amazing undergraduates work in my lab that have really contributed to some of the data that you'll see today as well. So I just want to acknowledge all those folks. And actually, I just want to point out the title, Collaborators, Colleagues, and Friends. I truly love being here because all of these are packaged into one. Is there anybody I don't like on here? No, I don't think so. No, they're all of them, which is great. So these are colleagues from different agencies from the university, other universities that I've worked with as well, and am really lucky for that. And some funding sources that have contributed in different ways to some of the projects that I'll present today. So I'm not going to spend really any time convincing you that drought is important. We see it in the headlines pretty much on a daily basis. This is just a few kind of headlines I've clipped. One about forest tree mortality and forest ecosystems impacting the species composition in those communities. Economic impacts, specifically thinking about range and cattle production. And this is more recently from Cape Town, running out of water, and so that's having a huge impact on not only the economy, but potentially public health in those areas. And so all of these factors and more, are important reasons that we are paying attention to drought right now. So we really need to understand what's going on during drought, how we can manage our resources in the best way possible to minimize those impacts. These are just a very few of the ecosystem processes, and I've slashed services because for the most part they're kind of one and the same. If you're an ecosystem scientist or an ecologist, you might think of them as processes. If you're outside of those fields, you might think of them more as services. So whatever you want to call them, these are just some of them. So forage production is hugely impacted by drought, and that's obviously got economic impacts. Wildlife habitat, as we think about conserving these natural resources and different animals, has a big impact. Water quantity, water quality, something we think a lot about here and see the impacts of drought on those services or processes. Air quality, I think I took out the picture of a dust bowl, blowing dirt around and having huge impacts, not only on the ecosystems, but on public health as well. And recreation, changing the structure of our communities might impact how we experience those resources. So these are just some examples to get you thinking about what's going on during a drought, what types of things should we be thinking about. I'm going to just talk about one of those real quickly, and this is kind of my bias and kind of where I come from scientifically that provides a backdrop, I think, a little bit for how I think in my talk today. But thinking about above ground net primary productivity. So I came out of labs that measured ANPP, thought about how that varied across space and varied across time. So I'll talk about both of those things real quickly. So this could be considered an ecosystem process, but if we think about it as potentially forage production or habitat, then we can think about it more as a service to animals, to humans, consumption, to all kinds of other things. So however you want to think about it, you can do it either way. So this is just a relationship to show you how ANPP varies across the Central Great Plains. So this is a spatial relationship between precipitation and ANPP. This was developed by Osvaldo Saldo when he was here. He got his PhD here, and so this was some work he did during that time. But you can see this really strong relationship across space. So if you go to a place that receives more precip, we get more productivity, or potentially more services, depending on what you're interested in. And really dry places are giving less productivity. This totally makes sense, right? None of us are surprised by this relationship. oops, I should have had one extra slide in here so you could see this a little bit better. This is in grasslands, but this figure here is across biomes. It includes coniferous forests and deciduous evergreen forests, just to show you that this relationship isn't just in grasslands, it's across all biomes, that we see this pattern and this relationship between precip and AMPP. And so these differences we see between these sites, those across space, really reflect differences in the amount of the resources, but also the species that are growing there and how they utilize those resources. So in forest ecosystems, we have trees that are functioning very differently than grasslands. That's the main take-home message there. But to get you thinking about interspecific variability, or how species differ in their ability to grow and respond to different soil moisture conditions. So then I also want to talk about temporal patterns. So this is thinking about within a site, from year to year as the amount of precip changes, how is the productivity of an individual site going to change? And we see a similar relationship. This is on Konza Prairie in eastern Kansas. We have the precipitation that falls in any given year, and AMPP on the y-axis here. And you can see, again, another kind of fairly strong relationship between how much precip falls and productivity. I'll just briefly point out here that this relationship within a site is usually much worse than across sites. And so really thinking about the interannual variability and understanding that becomes more important, because this just shows that it's not quite as simple as that spatial relationship. Okay, so that's the main things I want to say there. Also thinking about these temporal patterns and how much the biomass productivity varies from year to year. Really interesting kind of concept is that dry places are more sensitive to that variability in precip than wet places. We kind of like intuitively think that arid places have plants adapted to grow in dry environments, so they probably wouldn't be sensitive to changes in interannual or to interannual variability in precip. But we see the exact kind of opposite relationship. If we look here, we have the sensitivity of productivity on the y-axis, and the, oh, yeah, thank goodness. I was like, did I grab the wrong figure? Kevin's here, this is from his paper. Mean annual precip on the y-axis here. And so those places that are in drier climates have greater sensitivity to that variability in precip. So like, what's going on in these dry systems that they're so sensitive? This is another thing that we really need to figure out to understand how these different systems are going to respond to drought and how we might manage them. So AMPP, again, is this one ecosystem process. It's really nice because it integrates lots of processes. So it integrates what's going on in the individual plant and how it's responding physiologically to changes in the environment. It incorporates changes and kind of the amounts of nitrogen cycling going on in the system. Microbial activity, timing of those weather patterns. It might incorporate legacy effects depending on how you're analyzing it. So there's lots of things going on in this system that AMPP can kind of like incorporate and integrate. But there's lots of different things going on, and so we need to understand all of those to figure out what's going on. And so I am a plant ecophysiologist. I really focus on plant level responses to changes in the environment, and so that's kind of where I'm focusing. And this talk will focus on plant level responses, both interspecific responses, so how different species might respond to changes in precip or to drought, and intraspecifically. So if we look at the same species across different populations, are we seeing the same response within that species, or different populations responding differently? So I'll focus a little bit, or talk a little bit about both of those factors. All right. What did I want to say here? Oh yeah. So this is back, I showed this just a little bit ago, the different ecosystem processes that you might think about with drought. And I'm going to be focusing on these top two. Kind of thinking about forage production and wildlife habitat. And that's primarily what I've been thinking about recently. So you're getting kind of my recent snapshot of what mindset I'm in now, and I'm fairly stable. So hopefully it comes across as being clear. But that's kind of like my bias right now as I present this to you. So if you're like, what the heck is Troy talking about? Think back like, oh, he's thinking about kind of grasses and forbs primarily and how they're responding to drought. And this brings up an important point, in that many of us I know several of us in this room have a grassland inferiority complex. And you see it when you go to conferences and you hear a grassland ecologist talk, because the very first thing they point out is how important grasslands are in the global, on the global terrestrial kind of ecosystem. And so we use a pie chart kind of like this very loosely. We like to interpret it loosely. So we like to say that roughly 40% of Earth's terrestrial surface has ecosystems dominated by grasses, I think is the accurate way to say it. And so we do that by saying, oh, we've got 13% of the terrestrial surface occupied by temperate grasslands. Savannas, they're mostly grasses, really. And so we can add that, so that's 23%. And you know, most of these deserts have a large component of grasses as well. So now we're pushing 40% if we include those. And maybe we would even take some tundra if we're really feeling liberal on a certain day. So anyway, the point is that grasslands are really important. They're a large component of the terrestrial biosphere. And so how they respond to drought, I think, is vital for us to understand. The reason we have this inferiority complex, though, is I just did a super quick search on Google Scholar, and I put in the term forest drought, and I got 1.4 million citations. Who knows what those are? I didn't look through all of them. And I typed in grassland drought, and I got 0,400,000. So a whole million less citations on the impacts of drought on grasslands. So they're not well studied, although they're well represented across the terrestrial biome. So that's my grassland inferiority complex justification. All right, so why are grasses, I guess, so interesting will be kind of my next point. Or why do they differ from trees, and why do we have to study them differently? Or do we? I guess might be another question. But I'm going to try to convince you that there are different growth forms that we need to think about a little bit differently. And so really my talk will be in large part about kind of presenting some of those differences that we need to be considering. I'll show some results related to part of what we need to be considering, and kind of highlight some areas we're working on to fill in the rest of that kind of knowledge gap, if you will. But for those of you that think about trees most of the time and not grasses, this is just a picture of the crown tissue, or the tissue of the grass that's generally located just below the soil surface. And when grasses grow, they produce buds on this crown tissue. And those buds can then resprout and grow into new stems or tillers above ground. And so this ability of grasses to resprout after different disturbances is what makes it a really kind of powerful growth form as we look across the globe. So places where we have fire, it can kill the above ground tissue, but our buds remain safe below ground and resprout. Drought, they can senesce their leaves and resprout from these buds. They can be grazed and resprout from those buds. So it's that resprouting ability that makes them unique. And it's what we have to focus on when we think about how grasses are going to respond to drought. Because following a drought is resprouting from those buds is what allows that grass to recover and produce new forage, new biomass. My question to you, forest people, are do trees resprout after drought? And that one's not a sarcastic question. I've got another sarcastic comment for you later. This one's not. Because we know trees resprout after fire. Every citation I find on resprouting trees is about fire. So if somebody knows one about drought and resprouting of trees, I'd love to see it. I've looked for it, and I personally can't find it. So I'll put all of you on that task to try to help me figure that out. Okay, so I'm going to give you a really brief kind of summary of a grass life cycle. And I put this in air quotes because it's not really a life cycle. It's really just thinking about the vegetative reproduction of grasses. And so if we start and we think about a grass growing out on the landscape, we've got some leaves growing above ground. And these leaves are doing their best to photosynthesize and grab CO2 out of the air and incorporate that carbon into new tissues. Maybe they're going to store that carbon to utilize later to support the metabolism of the plant. But whatever it uses that carbon for, it's working really hard to grab that carbon when it's got those leaves above ground. And one of the things it's doing while it's growing is it's producing buds. All right, so I just showed you a picture of those buds. This is my much better drawing of buds. And so we've got this kind of bud production phase here. So if these leaves can photosynthesize well and get enough carbon to produce lots of buds, that's probably a good thing for the plant, right? Because it's got lots of potential to resprout after disturbance. We've got a senescence phase here. That could be triggered by lots of things. Or we could just think about it as the loss of the above ground tissue. But essentially, we don't have any leaves after this phase, and so we have a plant with just its buds and its roots sitting below ground. And whatever happens during that period of time when there's no leaves above ground, those buds kind of have to be self-sufficient, right? They're kind of isolated by themselves. So whatever carbon has been stored below ground to support their metabolism, that's all they have to work on. They need to stay hydrated in some way, so how are they staying hydrated is a little bit of a question. And so if we think about those drought conditions, we're mainly thinking about, can they support metabolism during that drought, and can they stay hydrated during that drought period without kind of the access of the physiology of the above ground tissue. And if those buds survive then after those conditions have been alleviated that caused that senescence, so we'll think about drought today. So we've got a drought, these buds survived, it rains, those buds can then resprout, grow new tillers, and then we can kind of continue this cycle. So very coarsely thinking, this is what's going on in a grass. And it's a little bit different than thinking about a tree. So I'm just going to divide this. What did I just do? Oh, I muted it. All right, so I'm just going to divide this in two sections for us to kind of like break it down and think about it a little bit. But I'm going to divide this right-hand side into kind of thinking about above ground processes. And so this is really thinking about the growth of those leaves and their ability to gain carbon and produce those buds. So that's what I'm thinking about during that period of time. And we're going to talk a little bit about how plants might vary in their ability to do this. And then we're going to have a below ground phase. And so this is thinking more about how those buds survive those drought conditions and have that ability or potential to resprout once those conditions have been alleviated. So I'm going to think about it a little bit this way. And then I just want to provide a little context in why I'm even interested in this and why grassland ecologists in general are thinking about this. So we're all familiar with the Dust Bowl, and John Weaver, who is down in this pit, was a grassland ecologist at University of Nebraska. And it was a horrible event to live through, but he learned so much about grassland ecology during that time. And he's studying the roots of prairie plants in this system, and he developed a map from that. But what he observed is through being out in the field during that time is once that, he referred to it as the Great Drought, once the Great Drought had been alleviated, new grasses were growing up, but they weren't growing up from seed. They were growing up from those buds that had been stored below ground. So this is the greatest drought that we have in our at least written history in the US, and some of these grasses didn't care. They grew right up after the drought was gone. Okay? How did they do that? We've got pinyon pine dying after 1 year of drought, and these grasses are surviving decades of drought. Sorry, that was my sarcastic comment about trees. So. So how did they do that? That's the question, and we really don't know. So we did a little one-credit class recently trying to dig up literature related to this, and really we didn't find anything of significance to help us really figure out what's going on. So that's what I've become much more interested in recently, in trying to focus a little bit below ground on these buds. So another context slide. That's just one biologically, ecologically, this is really interesting. But then thinking about more management oriented, and I'm thinking mostly rangelands right now, is this can be really helpful in thinking about adaptive management and making management decisions during a drought to enhance the sustainability of a ranching operation or the plants in the ecosystem. So I just have two examples here. If we have our plant again that had all of its buds below ground and all of them survived, once that drought has been alleviated, we've got lots of potential for new leaves to grow up and a lot of productivity. But if during that drought 4 out of those 5 buds died and only 1 survived, you have a lot lower potential for productivity. You've only got 1 bud to produce new leaves and grow. So this has a huge impact on what the productivity in the ecosystem might be after a drought, and this has been referred to as legacy effects. Osvaldo Sala, who's at University of Arizona now, or Arizona State, refers to these as legacy effects, that what goes on during the drought can impact your productivity afterwards. But again, we don't know what's causing these buds to die or experience mortality during the drought events. All right, so what did I say here? Oh, I think I'm trying to summarize. I don't remember this slide, so we're going to move on. I'm sure it's not that important. All right, so this is just kind of conceptually what I've been thinking about in considering what's happening to those buds during a drought. So how do buds survive these drought conditions? So we've got bud survival here, and it's relying, I think at least in my mind, on two major factors. One, I've talked about carbon already. So we don't know how many buds are formed during good years or even like slightly droughted years. And so understanding how many buds are actually formed prior or might be there prior to a drought is something we don't have a great handle on. How much carbon do buds need to survive? There's a model that kind of estimates how much carbon a bud might need to survive different lengths of time, but it's based on many, many assumptions, and we don't know if those assumptions are true or not. So we don't know how many buds are even going to be below ground under different conditions, and we don't know what they need to survive, and that's all primarily related to carbon and the growth of that above ground tissue. All right, so let's focus a little bit on above ground. So if we're thinking about survival of buds and thinking about water, how much water do they need? I mean, these buds are really, really tiny, but they're in a really dry environment during a drought, which has a really strong evaporative gradient, trying to kind of pull water out of those buds. So do they lose water during a drought? We don't know. Probably some, but we don't know how much. So thinking about how much water they actually need to stay hydrated, or I didn't even put this question up there, we don't know if they need to stay hydrated. There's plants that can dehydrate. They can lose all their water and recover. Can buds do that? We don't think so, but who knows? We don't actually know what level of dehydration a bud can handle before it actually dies. Then the question is, if it needs some water, where's a bud getting that water from? So you've got a bud that's all by itself out there on the crown tissue. Where's it getting its water from? Is it pulling it from the rest of the plant? Where's that plant getting it? Is it getting it from the soil? We're in a drought. Is there any water around? We don't know how those buds are actually getting that water. So I'll talk a little bit about kind of some of those factors and how buds might survive drought. And I shouldn't say we don't know any of the things. We have some ideas about all of them, right? So we'll talk through those a little bit. The other factor that we also have to consider, and I like to ignore, and plant people probably like to ignore as much as possible, is the biotic factors. Who's munching on the buds during a drought? All right, are there lots of organisms that want to utilize that fairly energy-rich resource of a bud? Are they utilizing those buds more intensely during a drought, less intensely? So we don't know how important this factor is either. I'm not going to work on that, or I'm not going to talk about that today. Maybe I'll work on that. We'll see. All right, so these are about the same questions, but I want to group them into this above ground and a below ground. So when we think about the above ground tissue, and we're talking about growth of that tissue, we're talking about primarily carbon. How much growth occurs in that above ground tissue? How much carbon do they store? How many buds do they produce? That's what we're thinking about. And then below ground is thinking about how much carbon do the buds need, but then thinking primarily about staying hydrated and whether they need to stay hydrated or not. All right? So thinking about above ground growth, and why we really need to think about this on grassland ecosystems and interspecific differences is that in grassland ecosystems, they're highly biodiverse in their plant species. So you can get many hundred species. It depends on like what we call a grassland, right? But we've got like 100, 200 species on grassland ecosystems. On Konza Prairie in eastern Kansas, there's more than 200 species. So they're highly diverse as far as the plants that are there. And when we get a drought, not all of those plants are going to respond the same way. So we need to understand how different plants in those communities are going to respond, so then we can kind of make predictions about if we get a drought, what species are going to be gone from the system, or which ones are going to be like producing more or less, and quantifying that type of information. So that's why I'm really thinking about these interspecific differences in above ground growth. So I'm going to walk you through some of the results, and I'll tell you now, and I'll show you another slide related to it, but most of the work we've been doing in my lab is related to this above ground growth. And starting to think about this bud kind of survival has been something that's more recent. And so I'm going to show you quite a bit about what we know above ground and then think about what's going on below ground. So this is one way that we quantify how plants are responding to drought. This is a leaf level measurement, and I'll walk you through it, kind of what's on the graph here, and then what the metric is we actually use to quantify this. So on the y-axis here we have the percent of maximum leaf hydraulic conductance. All right, so if you think about plant growth, you all know that plants utilize water. And if they're photosynthesizing, they're using water. All right, those two things are linked very tightly in plants. And so if plants can't actually move water through their tissues, they're going to have to stop growing. All right, so this percent maximum leaf hydraulic conductance tells us the capability of plants to move water through their tissues. At 100% means they're really healthy. And as plants dehydrate, which is shown to the right on this axis as leaf water potential, as they get more dehydrated, they lose the ability to move water, and that's going to cause their growth to slow down or potentially stop. So what we do is we create these curves on leaf tissues, or you can do it on stem tissue. This data set here was, I like to point people out, I hope that's okay. Robert, who's sitting in the back here, he created this data set measuring leaf hydraulic conductance as those leaves dehydrated. And we pick the point where the plants lose 50% of the hydraulic conductance, and we call that P50. So that's what's here. All right, so P50 gives us an estimate of how plants can tolerate, or how well they can tolerate, drought or drying soils. If they have a P50, so more negative values of P50 means a plant is drought tolerant in its leaves, or it can keep conducting. Less negative values means it kind of shuts down really quickly as the plants start to dry out. All right? So if you're not familiar, leaf water potential, they're all negative values. So that's why we have to deal with negative values. So my apologies. I hate negative values, but anyway. So I'm just going to show you a couple data slides to show you why this P50 value is important in understanding plant function. This is some data that I was lucky enough to go to China last semester, and so we looked at bamboo, which I've always wanted to do a study on bamboo, so we got to do bamboo, and then another tropical evergreen broadleaf tree while we were there. And so we generated the same types of curves, and this is what you see here. We've got hydraulic conductance on the y-axis and leaf water potential on the x-axis again. One thing that I want to point out here is the red and blue symbols show the ability of a plant to recover from a loss of hydraulic conductance. So a red point that has an increasing arrow on it means that that plant was suffering, but when you gave it water again, it could recover and maintain its growth and recover its growth. And a kind of horizontal or downward trajectory arrow means that plant couldn't recover from that particular condition. So what's interesting here is so P50 is the metric I just gave you when a plant loses 50% of its hydraulic conductance. We also calculated a P88 is when a plant loses 88% of its hydraulic conductance. Mainly, the main take-home message is when a plant gets beyond P88, it can't recover. We don't really see any kind of recovery. That kind of means death for a plant. All right? This data set is similar, but what we did is we actually were able to optically detect the decline in conductance in those leaves. So we had a method for seeing like how kind of continuous the pathway of water transport was versus when it was not filled with water anymore and when it was air filled. It turns out that when plants become air-filled is really correlated with that P88. So when plants lose 88% of their conductance, all of the pathway that should be filled with water is now filled with air, and that, we think, prevents their recovery from that kind of loss of hydraulic conductance. So this metric tells us something really important about what's going on in a plant, and it gives us a really nice metric to say, like, okay, when it hits this point, this plant is probably not going to recover, and it might be on its way out, or at least this particular leaf, okay? So we think this metric is really important kind of like physiologically. I'll show you some data slides and how it correlates with the distribution of different plants as well. This is P50 value is here on the y-axis. The higher on this y-axis, the more negative the values. So this actually means plants up here are more drought tolerant, plants down here are less drought tolerant. And then we have the fifth quantile of the temperature of the wet quarter. We think what this is telling us is how hot it is when it rains. Okay? Places where plants are growing during the hot season or when it's fairly warm, those plants have greater drought tolerance than plants that are growing when it's in cooler season. So this is probably telling us something about like dry season grasses versus cool season grasses. It was a little ambiguous, so we're not 100% sure. But then we got some other data that kind of supports that idea with slightly different metrics. This was all C4 grasses. This is a combination of C3 and C4. So we have precipitation of the wet quarter in the 5th percentile. Again, it just means the driest places that a plant is found. All right? And then we have osmotic potential on the y-axis. So I'm changing it up for you. But osmotic potential and P50 kind of tell us the same things. All right? They're highly correlated. So we've got, my apologies, we switch our axis here. Plants down here are more drought tolerant than plants up here. So plants that tend to grow in the driest places have those leaves that are more drought tolerant. This is totally intuitive, right? We kind of like expected this all along. We kind of knew this. But now we have a metric to actually provide some quantifiable measures of those differences between the species. So, all right, so summary up to this point and thinking about this above ground growth. And so we see a lot of variability between species, even within a plant community in this P50 value, or in this metric that tells us how drought tolerant leaves are. We see, so this trait, this P50 value, correlates pretty well with the distribution of species across the landscape, as well as the plants within an individual community. So it seems to be a fairly useful trait in understanding how plants might respond to drought. The main thing to think about is that a plant with a more negative P50 value is a plant that's going to be able to keep growing longer into a drought or under drier conditions. So if we're thinking about a plant producing buds and storing carbon for those buds to survive a drought, a plant with more negative P50 values is probably going to be able to do that better under dry conditions than a plant with high P50 values or less negative P50 values. So this is how this kind of starts to tie in with thinking about our bud survival. So we can start making some predictions now. Plants with more negative P50 values will grow better during a drought, produce more buds, and store more carbon. That's our prediction right now. Very simple prediction, way overstating the very little data I just showed you. I just want to point out this. This is a paper that Robert and a bunch of graduate students across several different departments at the university just got accepted in functional ecology, looking at what traits are people measuring to understand plant responses to drought. And our hydraulic traits are kind of third on this list, and less than half of the studies have utilized that relative to kind of broader morphological traits. So this is just to highlight, one, we think this trait is really important in understanding drought responses of plants, and we're not measuring it enough. We need to measure it more and figure out how it's really, how it can really quantify plant responses to these conditions. Okay, so now I'm going to get back to our strategies a little bit that you guys discussed before I started talking. So these are two different strategies of drought resistance. And I'm going to take those strategies, like Miranda said, but I'm going to apply them to tissue level. I'm thinking about, we could think about a leaf on a plant and how it might respond to a drought. So a drought-tolerant plant is going to be a plant that can allow its leaves to dehydrate a little bit and still maintain function and keep that leaf on the plant. So you can think about a leaf being dehydrated but still functioning. Drought avoidance is a plant that wants to keep all of its leaves really well hydrated in order to grow. And if it's not hydrated, that leaf isn't going to function. And so a plant might avoid drought in its leaves by having really deep roots or accessing groundwater. Or maybe it totally shuts off transpiration and it can isolate it from its environment. Whatever the mechanism, it's trying to keep the hydration status of its leaves high. All right? So there's some different strategies for how plants are going to survive in a drought. And so we were interested in seeing how much variability there was in an individual species in kind of like whether all populations utilize the same strategy or not. So this is some research that Julie Bushy did. She was a master's student that graduated last summer. And she collected Bouteloua gracilis samples from New Mexico up to South Dakota and got a large gradient. And then her and another student collected them from kind of here in Fort Collins up the Rockies, the Front Range, to get an elevational gradient as well, and to see how these plants are responding to drought. And our prediction was those plants growing in New Mexico, in southern Colorado that experience really dry conditions, they were going to be able to tolerate drought well. And when we exposed them to a drought, we probably wouldn't see a big reduction in their growth. Can you guess what the results were based on how I set that up? The exact opposite, right? So, so what we have here, I'm going to highlight this graph down here. This is leaf water potential on the y-axis. More negative values kind of you typically indicate a plant that's a little more stressed than high values. And then we have the growing season aridity for where each population was collected. Those on the left-hand side are populations from more arid environments, and the right hand are the populations from more mesic environments. What we found, oh, and one other thing, the dotted line shows plants that were grown with abundant water, so they were watered every few days, had plenty of water. Plants represented here along the solid line, those are plants that were droughted, were given a limited amount of soil moisture while they grew. What we see is plants from the mesic environments, they were able to keep their leaf water potential at the same level. So they didn't change their leaf water potential at all, indicating they didn't really experience that much stress. The plants from the drier environments, however, had a more negative leaf water potential, so they were experiencing drought stress a little bit more. So this kind of like highlights differences in strategies. These plants are allowing leaf water potentials to drop, maybe like a drought tolerant strategy. These are keeping them the same, maybe a drought avoidance strategy. And the way they did that is plants, this shows the osmotic potential, which is basically how many sugars are in the cells of the plant. Plants from the mesic environments actually increase the amount of sugars in their cells, and that helped them stay hydrated during those drought conditions, where we didn't see that really happening at all in the plants from the drier places. So we definitely see some evidence of different strategies within the same species across these populations. It also turns out that that impacted then their sensitivity of growth to these droughts. So on the bottom line here, we just have the sensitivity of biomass production, both above ground and below ground, for populations across that same aridity gradient. And what we see is the plants from the driest places were actually the most sensitive to the drought compared to the plants from the wettest places. So it turns out in this situation, being a drought avoider by kind of adjusting the sugars in your cells, it was actually a benefit to these plants, at least under this drought condition. All right? Interesting, oh, I didn't put it on here. So I showed you earlier Kevin's data about sensitivity of ecosystems to precipitation, and that the driest places were the most sensitive. This actually kind of, even though it's within the same population or within the same species across populations, we see a similar pattern. So something interesting going on here with plants growing in arid environments. And kind of suggests maybe we don't quite actually know what's going on with them as much as we thought. So adding to this summary, I already talked about the first two. Next one here is populations differ in their drought resistant strategies. We see some maybe trying to be drought tolerant, some being avoidance. But we think we can kind of quantify this variability looking at osmotic potential is one measurement that keeps coming up in a useful way in kind of describing these different strategies. So although we have this variability within a population, we can quantify it, we can measure it. All right? So to kind of like modify and build on our predictions a little bit, we have a little more confusion now. The first data sets I showed you made it seem really simple. Now we've got this plasticity within a species that we have to account for too. So we might say that plants with more negative P50 values will grow better during a drought, but if there's plasticity within that species, we need to account for that when we look at community level responses to different drought events. All right, So those are kind of thinking about interspecific differences, intraspecific differences. Now I want to think about individuals growing across a growing season, or that's kind of the way I'm envisioning it. And in this scenario in my head, oh man, that scared me. Because we usually start on the hour, and I'm like, I have only been talking for 13 minutes? All right, that makes more sense. All right, so temporal growth patterns. So I'm thinking about a system that's starting with a decent amount of moisture and it's drying out over the season, and thinking about how these plants with these different traits might be growing in those systems. So back to this P50 value one time. This is P50 of that hydraulic conductance. So plants on the right are more, we're calling more drought tolerant. Ones on the left are less drought tolerant. I also calculated P50 as stomatal conductance. So what this data is basically telling us is that plants are shutting down their stomates before they reach their P50, because all of those data points are below the 1-to-1 line. So that's telling us plants are trying to prevent this kind of catastrophic event I showed you earlier with reaching this P50 or potentially this P88. So as we think about soils drying out, we know that plants are closing those stomates as the soil dries in order to prevent this event from happening, or crossing this threshold, if you will. The other thing, though, that's interesting is it's not just P50, of course, we all knew this, that there's more to it than just what's going on in the leaf. This is combining the P50 value with some information about rooting depth. And I'm going to focus on the right panel here. They're telling us similar information. And what I was getting at with this kind of analysis is thinking about how long of a growing season does a plant have based on its ability to tolerate drought, or this P50 value, which is down here. Oh, I switched it to psi. I'm sorry. It's the same thing, P50. And also then think about rooting depth. So my initial hypothesis would be, based on previous data, is that plants with more negative P50 values, more drought tolerant, they'd be able to grow for longer in the season, right? As the soil dries, they don't care as much, so they keep growing. But when we think about and incorporate where they're getting their water from, it kind of changes the story. So these black symbols here represent plants that were getting their water, and their roots were all kind of isolated in the shallow soil layers. And we see that pattern that I just kind of mentioned. Plants that are more negative, they're going to be to the left side of this axis here, so more drought tolerant. They were able to keep their leaves on till later in the growing season. This is the day of year when they had 100% leaf senescence. All right? So they kept some of their leaves on to day 260 about, when they were really drought tolerant. Plants with intolerant leaves were dropping their leaves much earlier in the growing season. So if we think about a plant's ability to produce buds and store carbon, in one system, these plants are going to have an advantage in that system. They might be able to produce more buds, or at least store more carbon to support those buds during a drought, whereas these plants might be more susceptible to not being able to do those things and not being able to support those buds during a drought. But if plants that have deep roots are represented by the gray symbols here, they have the exact opposite pattern. And I'm not sure, these plants are kind of in between as far as rooting depth. So I don't want to focus on or suggest this is a linear relationship. But if we kind of focus on all these data points up here, we see that plants with intolerant leaves, or they have really less negative P50 values, they can keep their leaves on just as long as the drought tolerant ones is if they have deep roots. So you can kind of have, you can have one or the other. We don't know if you can have both. So the big kind of next question is, are there plants out there, and the answer is probably yes, some, that have both deep roots and drought tolerant leaves? But we need to figure out if there's a trade-off or not, so we can understand what different strategies plants might be using in a plant community. Okay, so now adding to this summary slide a little bit, we've talked about the first three. So this last piece of information is that plants that utilize water from deeper depths in the soil don't necessarily need to have drought-tolerant leaves. All right? So they can have intolerant leaves, and they can still have a really nice long growing season to accumulate carbon, produce as many buds as they can. All right? So we have to refine our predictions a little bit again. Plants with more negative P50 values will grow better during a drought, except for those species that might utilize deep water. All right? So now we have to incorporate that. Luckily, well, we can kind of estimate rooting depth. We're working on trying to do it better. Sometimes it works, sometimes it doesn't. You could go dig them up, which is you disturb the system. You get a lot of information about biomass, but you don't know where the plants are actually getting their water from. So we're trying to work on this technique and develop it a little better because obviously it's really important. I just want to say one thing about, actually I'm going to skip this for sake of time. And if we have time, we'll come back to it. So what I've told you, everything I've shared with you, well, first I shared with you all this really cool framework for how plants are like surviving drought and regrowing. And what I've shown you might be like this much of a piece of the pie. It's just what we've learned about above ground tissue and thinking about how plants might accumulate carbon and produce buds. But we have this whole other piece of the pie that we don't really know about. And so that's what we're kind of working on now. And so I just want to highlight some of the things we're trying to work on to figure out what's going on as we shift from the above ground production to below ground production. So this is what takes us to kind of next steps. We know what to measure, or we have a pretty good idea what to measure for thinking about above ground plant responses. So we can begin to make some predictions, trying to incorporate that information in models that might predict plant growth responses on an ecosystem level. So now we need to shift our focus to the below ground and thinking about these questions. These are the same questions I showed early on, so I won't go over them right now. But I just want to highlight a few projects and what we're doing to try to answer these questions. So Scott Bradfield is a PhD student working in my lab. He's looking at the below ground responses to droughts of varying severity at the shortgrass steppe. Here's him like kind of fondly looking at his shelter that he's created, proudly like, yeah. But he's going to drought plants on the shortgrass steppe for different periods of time and see how many buds survive, how many new leaves can regrow after that drought has been alleviated. So really starting to directly test some of these questions that we have. We also have a project started that is looking at the effects of fire and drought on the below ground responses of plants and how they can regrow after a fire. Fires have the potential to heat up those buds, and so whether those buds can survive high temperatures or not is a question that we're directly asking. And then we want to look at the effects of drought as well on those buds. So again, starting to directly look at the impacts of the environment on those buds and whether they can survive those conditions or not. Oh, I didn't really mean to do a video. It's just like what I grabbed real quick. And then this is one of the undergrads in my lab that helped. This is Tanner. He graduated last year. But Tanner helped with some preliminary data on a study where we actually were able to take buds from grasses, expose them to different osmotic solutions, which kind of mimicked drought. And then we measured how many of those buds grew out into new tillers. And so we're still measuring and sampling a lot of that information. But this is just showing us we have 3 different species represented by those different colored lines. So we're definitely seeing species level differences in how they can tolerate and survive these different drought periods. So we're really excited to start digging into this a little more and see what's going on across these different species. And then we have a project that we're trying to get going. It's mostly going up on Thunder Basin National Grassland, where we're, I wish these pictures showed up a little bit better, but we're increasing the interannual variability in precip. And so we have pairs of plots, which you can't really see here. We have one plot where we're capturing 50% of the rain, and then we're actually pumping that water onto another plot. So this plot will be dry, this plot will be wet. The next year we'll flip them. So the dried plot will get wet and the wet plot will get dry. And so we're going to look at species level responses to that interannual variability and try to understand how like rooting depth and these P50 values of the leaves will impact the growth of these different species. So that is all I have. Thank you for coming and being interested in my research. Hopefully you were interested in my research. And so I will take any questions now that you've got.
So for that last experiment, are you going to measure the traits within the plots each year, or are you going to just assume the trait is constant and apply it to the...
I love making bad assumptions. No, we'll measure them in each plot. I don't know if we'll do it each year. We'll have to decide and see how much time we have. Yeah, but that's a good question. So, yeah.
I assume a lot of what you're looking at is a lot of lab work. So I assume a lot of these watering experiments are going to be kind of a constant application of water. Do you look much at like intra-annual variability in water?
I have not done much. Alan Knapp's lab and Mindy Smith's lab does a lot more of that, looking at that interannual variability. Kevin and Dave, who are both sitting here, have done some of those types of experiments too. So corner them afterward and ask them more about it. But yeah, certainly it's showing up as being really important, and when that rain comes, how long the interval is between different rain events is really important in that productivity. Okay. Oh, Kevin. Yeah?
I'm curious about phenotypic plasticity. What if you took the ground from the music environment and planted them in the arid environment? Do you think it's genetic or
I think, well, it certainly seems genetic because this was a greenhouse, so it's kind of like a common garden study where we saw those differences. I do think it'd be really interesting to maybe manipulate. So what we think maybe is going on is the plants from the New Mexico area, they're adapted to utilizing the monsoonal rains and then having really long drought periods in between. Whereas the plants from further north have more consistent rain. And so they just are adapted to those different kind of conditions. That's kind of our hypothesis right now. So we'd like to manipulate the rain patterns in each of those systems or across that gradient and see how the plants respond. But the thing that jumps out at me in thinking about that as we're thinking about where to get our seeds from in restoring systems, we often think about, oh, from a drier system might be better, but there's certainly thresholds you might cross that would lead to having a plant that has the wrong strategy for the system you're planting it in. So I think when we're thinking about like restoration, what seed sources to use, it'd be useful to have more of that information and understanding before we make some of those decisions. Tony.
Would you say that the mechanisms and the traits in the kind of vascular plants you're studying are the same with wheat?
Yeah, so I didn't show any of that because I was trying to minimize trees and build up the grasses. So, but really, so to be honest, like most of what I've done has been like, hey, that works on trees. Are grasses the same? And so a lot of these traits that are showing up as important are also important in forest ecosystems as well. Both like on what species are surviving drought as well as what species tend to grow in drier places versus wetter places. So it tends to be fairly consistent for that trait. Yeah?
If I could follow up on that second to last question. What is the trait or the adaptation that you suppose could be going on? Is it root architecture?
No, we just think so based on some other data that we have, the populations from New Mexico actually have really high photosynthetic rates, higher than we kind of expect. And so we think what they're adapted to is kind of surviving long periods of drought. When they get their rain, they grow really fast and then shut down again. And so they're not really adapted to trying to grow during moderate times. It's either, you know, go all out or don't go at all. Does that make sense?
Yeah.
Okay.
During the time, I think we're going to end up in one question.
That was great. Yes.
So are you guys planning on doing any biochemistry work in the future?
Scott is going to be doing some. I try to stay away from chemistry as much as possible. But yeah, Scott's looking at some of that. We're really interested in, well, yeah, this might be kind of beyond what you were thinking, but so looking at some of the proteins especially, which isn't exactly probably what you were thinking about, but kind of what compounds and hormones are released during those events, and like what's going on in the buds that might protect them from those drought events or allow them to survive that might not be some of the other mechanisms we've hypothesized. So, yeah.
Okay, so let's wrap up. Thank you so much for...
Thank you.